Hello, everyone. Thank you so much, Eva, for the kind introduction. And thank you also to Twist for inviting me to speak today. So again, as Eva already said, my name is Stefania Zingale, and I am currently a PhD student at the Institute for Mommy Studies at URAC Research in Bolzano in Italy. And in this talk, I will give you an overview on our project Cells in the Alps, the cells up and down the Alps, and I will share some of the work I've been doing for my PhD, which focuses on the genetic investigation of late Iron Age individuals from both north and south of the Alps. So before I dive into the project itself, let me briefly introduce who we are. So URAC Research is a research center based in Bolzano in northeastern Italy. And within URAC, our Institute for Mommy Studies was founded in 2007. And since 2018, we've been based at the Neu Tech Park, where we have our offices and our laboratories. So here we have our offices and our laboratories. So our institute focuses on studying ancient biological remains, both mummified and non-mummified, using a range of approaches. So we have three main laboratories. So in the conservation lab, we study the preservation state of mummies, and we develop innovative conservation methods tailored to each case. And in the bioanthropology lab, this lab covers not only the reconstruction of the biological profile of an individual, including also paleopathological analysis, but here we perform also collagen extraction for stable isotope analysis and apply techniques like CT scans and 3D reconstructions. And finally, we have a laboratory for DNA analysis. We have an ancient DNA lab and also a modern DNA lab. And here we study not only human DNA, but also we focus on metagenomics like ancient pathogens, microbial DNA, fungi, and oral and also gut microbiome, and so on. We are not a very big institute, but what makes us strong is our collaborative perspective. So our team brings together people from different backgrounds, and that mix of expertise allows us to approach complex questions from multiple angles. And so let's move to the main topic of today. So the Cells Up and Down the Alps project. So this project is a multidisciplinary project funded through a joint program between Switzerland and South Tyrol. So you can see we are a lot of people involved in this project, with the main project partners, the University of Bern in Switzerland and URAC Research. And so this project is funded by the Swiss National Science Foundation and the Autonomous Province of Bolzano. And our goal is to better understand the origins and the mobility of late Iron Age groups from north and south of the Alps. And we define north as modern-day Switzerland and south as northern Italy. And we do this by combining genetics, isotope, osteology, and archaeology. So why do we even need a project like this in the first place? So ever since working on this project, I realized that the definition of cells is not really straightforward. So in general, they are defined as groups that spread throughout Europe during the late Iron Age, so the 4th to 1st century BCE. And we associate with these groups the archaeological material culture Latin, which has its origin in the name-giving locality in Switzerland. And even though we can associate the cells to this material culture Latin, at the same time, the occurrence of this material culture does not equal to cells, in the sense that not all archaeological findings associated with this culture are automatically defined as Celtic. So in general, the late Iron Age period, Latin, is characterized by the high mobility of the groups associated with this culture. And especially during the 4th and the 3rd century BCE, archaeological and also historical findings document a southward movement of this culture, in particular towards the regions of Italy, the Balkans, and Turkey. And this is actually what makes the area surrounding the Alps of such high importance when studying the genetic history and the mobility of these groups. So with this study, we want to contribute to the current state of research on Iron Age communities in Europe by trying to reconstruct the genetic history and migration of late Iron Age groups from the area surrounding the Alps, as I already said, which is a key point for the expansion of this late Iron Age Latin culture. And for the genetic part, we can tire 3 big research questions. So we want to map on a global scale the genetic diversity and relationships between the studied late Iron Age groups and other modern and ancient individuals from Europe. And to do this, we need to understand first the regional genetic diversity and structure of each studied group. And we're also trying to trace genetic links between them. And in order to accomplish these 2 research questions, we need to zoom in first on local dynamics. And we do this by studying biological sex, uniparental transmitted markers, and biological relatedness within each group. And the integration of isotopic, archaeological, and anthropological data. With this integration, we aim to gain insight into the social organization of these late Iron Age groups. So our data set includes 227 individuals from 27 different archaeological sites. So 119 individuals were excavated in modern-day Switzerland. So here is Switzerland, and they belong to several different archaeological contexts. You see all the different dots. And 108 individuals originate from modern northern Italy, and most of them belong to the Necropolis Seminario Vescovile, the blue rectangular. So for ancient DNA analysis, we mainly selected petrous bones, molars, and inner ear ossicles, since those skeletal elements are the ones that we know preserve human DNA the best. And in very few cases, if those skeletal elements were not present, we used long bones for DNA. And I show here a petrous bone before sampling and after sampling. So we just make a really small hole to get the powder we need for DNA extraction. The laboratory work was then conducted at dedicated facilities in URAC research and following strict ancient DNA standards. So after sampling, after DNA extraction, and after the construction of double-stranded, double-indexed genomic libraries, the samples were then subjected to a first molecular screening, like shotgun sequencing, in order to perform quality assessment. And the samples that reached at least 1% of human DNA, which is the threshold to include our samples in our analysis, those samples were then selected for the enrichment of more than 1.3 million autosomal SNPs. And for this enrichment, we used a customized kit from Twist Bioscience. And these enriched samples were then sequenced again, and the generated genomic data was then used to perform bioinformatic and statistical analysis. The generated sequencing reads are mapped onto 2 different reference genomes. So first, we map onto the human reference genome, HG19, and with this data, we can authenticate the DNA and check that it is really ancient and belongs to our individual. We we try to determine the biological sex of the individuals, and we approach the estimation of biological relatedness. And in this project specifically, we use 3 different methods on autosomal DNA that allow us to estimate kinship up to the 3rd degree. And for male individuals, we also analyze the Y chromosome to investigate possible paternal relationships. And so these steps basically set the stage for comparative analysis, so population genetic analysis. And here we apply methods like FSD statistics, PCA, like comparison to other data sets, and also model-based clustering like admixture or QP admixture, and this to explore the genetic makeup and diversity of the studied groups. And in parallel, we map also the reads to the mitochondrial genome, and this allows us, again, the authentication of our data. It allows us to estimate contamination and reconstruct also the complete mitochondrial genome. And since mitochondrial DNA is inherited only maternally, this gives us also insight into the maternal kinship patterns among our studied individuals. And yeah, let's have a look at how our late RNA samples actually perform. So in terms of quality, so the quality assessment of the shotgun data shows actually quite a good quality and authentic ancient DNA of our samples. Only a few individuals had very low endogenous content, so it's here only some samples, and these samples were then excluded from further analysis. And after applying the twist target enrichment, 178 individuals out of the 227 passed the quality control, so human endogenous content higher than 1%, and mitochondrial DNA contamination less than 5%, and for males, X chromosomal contamination below 3%. So these individuals were then included in the analysis I described earlier. The enrichment step really made a difference for our data set. So by applying the twist target enrichment, we were able to include many more individuals, especially in more downstream analysis, more specific analysis, than would have otherwise been not possible. So without the application of this enrichment kit, some samples would have been excluded because we could, for example, not estimate contamination, and or we don't have enough data in order to perform some analysis. So the kit allowed us to increase in general also the mean coverage on the whole genome, as you can see. So the red one is the shotgun data, and the gray one the target enrichment. So we have here an increase, and if we focus on the 1240K SNPs panel, which is commonly used for population comparisons, we see a strong increase in both here the number of SNPs of the 1240K data set, and also of the mean coverage of these SNPs. And the genetic data generated through this project has already been used into 2 multidisciplinary studies, which I would like to show you now. So the first case study I would like to share focuses on the necropolis of Seminario Vescovile in the Verona province. So we are here in northern Italy, so south of the Alps. And in this funerary context, the archaeologists uncovered 161 human inhumation. And interestingly, we find 16 inhumations that include animal remains. And among these 16, we find 4 co-burials that involve whole dogs and horse skeletons, so which are species that were not part of the human diet actually in this time, so something special. And to explore potential social or symbolic meanings of these depositions, we used a multidisciplinary approach, including zooarchaeology, stable isotope, ancient DNA, and radiocarbon dating. And interestingly, the results showed no consistent demographic, no genetic, dietary, or funerary patterns among the individuals buried with animals. For example, genetically, we identified 5 males and 7 females. This suggests actually no preference for either sex for this kind of depositions. And we also applied the 3 methods I told you before to test biological relatedness, and added also information from mitochondrial and Y chromosomal data, so maternal and paternal lineage. And none of these indicate kinship, at least to the 3rd degree. And this means that this possibly ritual status does not appear to have been inherited through bloodlines. But still, the presence of complete dog and horse skeleton points to some sort of symbolic or emotional significance. And one dog, for example, had a distinct isotopic profile, like it's really, really close also to the humans. And this suggests that this dog was raised or cared for differently, and perhaps it reflects also a special role of this dog in this community, or to the individual that it was buried to. So in summary, the co-burials at Seminario Vescovile seem to reflect actually a complex and not really uniform cultural behavior, rather than simple offerings or elite markers of these animals to these people. And now let's move north across the Alps to a very different but equally fascinating site. So the site of Cournot in Switzerland. So here archaeologists discovered the remains of a late Iron Age wooden bridge in Lake Neuchâtel. And alongside the structure, they found weapons, animal bones, and about 20 human skeletal remains, which were entangled in the wooden structure. So here, I guess you can see here. And this finding raised actually big questions. So was this a tragic accident, like a bridge collapse, or could it represent ritual or sacrificial activity? So we applied the same integrated bioarchaeological method, combining osteology, in this case also taphonomy, so studying perimortem bone lesions, isotopic analysis, radiocarbon dating, and paleogenomics. And with this study, we mark here the first genomic data set for Iron Age Switzerland. Really, really nice. And so for the isotopic data, so the isotopic data reveal several non-local individuals, like the individuals here below the lines and above the lines. These ones were all outliers. And this points to a high mobility among this community or this group. And we performed a genetic analysis on 11 of those individuals. And our analysis showed that all adult individuals, so we had 10 adult individuals and one subadult, and the 10 adults were males, and only the one subadult was female. And also with the Italian side, we tested for biological kinship, but we did not find any close biological relatedness between those individuals up to the third degree. And we did also a very qualitative comparison analysis. And this analysis revealed genetic affinities of those individuals with other Central and Western European Iron Age groups, which aligns to the area of the finding. And so these findings, together with the fact that we find only blunt force traumas, point to the likely scenario of an accidental collapse of the bridge, possibly while people were crossing it. And however, we cannot fully exclude a ritual or sacrificial interpretation either. But yeah, so in essence, Cournot reflects the complexity of interpreting bioarchaeological evidence, especially when this kind of ritual and accidental explanation may overlap. Yeah, and yeah, that's it actually. Thank you so much for your attention. And if you would like to know more about our project, please visit our webpage. And yes, thank you so much for your attention, and I'll be very happy to take questions.
Thank you, Stefania, for your presentation. Very insightful. We're really happy to have you on our webinars. We're now open for questions, so please type them into the Q&A box rather than the chat box. We already have a few questions, so let's kick off with the first one. Someone asked, how do isotopic and genetic data complement each other when reconstructing migration or kinship patterns in ancient groups?
Okay, sorry, can you repeat?
Of course. How do isotopic and genetic data complement each other when reconstructing migration or kinship patterns in ancient groups?
Okay, so both isotopic, like stable isotope analysis, and genetics are, when they're used together, they're really, really strong and powerful to understand migration. So, and also, of course, let's say the evolution of a group or of a population. So, a stable isotope analysis gives more an insight into what happened in a very short time. So, for example, we can understand if a person was born in one place and then died in another place, rather than was born and then died in the same place. Instead, the genetic part gives more like an insight on what happened in history with the group that this individual was living at. And yeah, they're both very strong together. Like, yeah.
Okay, thank you. Next question is, how do ancient DNA results help distinguish between mobility and culture transmission within the Latin sphere?
Yes, this is really interesting actually, because looking in genetics, you can, or the genetic profile of an individual, you can understand, or not understand, or it can give you a hint if the transmission is like an actual migration of this individual, or only like or a cultural transmission. So if you would find somewhere an individual that was buried with this typical Latin characteristics, but has like a very different genetic profile than anyone else, then you could also, like, this individual probably took over the this kind of culture. Yeah, I think.
Okay, thank you. One more question that we have is, what is the timescale of applicability for carbon dating?
Okay, I'm not really a pro on radiocarbon dating, but I think it's several 100,000 years. I would need to look this more up.
Sure, thank you. And one more question that we have, is the bone you use fossilized or mineralized?
It's not fossilized, so it's basically you can imagine, yeah, since it's not that old, it yeah, it's not like a fossil, it's not like hard as stone, but then this depends a lot on the where in which kind of ground an individual was buried. So I had some samples from Switzerland, for example, that they were like in the Alpine area, and the bone, like the environment changed the structure of the bone completely, and it was so fragile that at the moment that you had it in your hand, it was just completely like broken. And sometimes it can happen that you have like a really, really, really hard bone, that it looks like, it feels like a bone, yeah, a rock. So sorry.
And I think we have one more question, or last one. Your research extracted DNA samples from specific bone structures. What is the current time limit consensus for usable DNA extraction?
Is this more like, if we talk about like the time limit that we have, like the moment that we extract the DNA, so in the liquid, let's say, we don't really see a very high degradation the moment that we extracted the DNA from the bone. So we are also using extracts that are like older or from several years in our institute. If the question is about like the current time limit, let's how old the DNA can be in order to perform like a proper DNA extraction, then it really depends. I heard that several million of years you can still do DNA extraction, but I think this really depends on the preservation of the of your samples. And I think someone answered the question about the C14 dating. I feel really bad about it that I didn't know that 50,000 to 60,000 years.
All right. Well, thank you. I think that was our last question. So thank you all for joining us. And thank you again, Stefania, for sharing your work with us.
Thank you so much.